Okay, so welcome back to this next video in which we are discussing synaptic vesicle exocytosis and endocytosis. Okay, right, so we have now discussed synaptic vesicle exocytosis in full. We've also discussed how you actually break apart the cis-snare complexes once full fusion has completed, okay, and that's through the NSF protein bound to its cofactor of SNAP. Okay, and it hydrolyzes ATP to ADP and inorganic phosphate and uses the energy from that to break apart the snare motifs of these three snare proteins and break them apart into three separate snare proteins once again. Okay, right, so now what I want to turn my attention to is endocytosis. Okay, and the process that we're going to study is Clathrin-mediated endocytosis. Right, so this is the process by which we are going to re-pinch off that portion of vesicle that we've just fused with the plasma membrane. So let me just draw this out. So let's say this is our axon terminal here, and let's say that this portion of the plasma membrane here in purple, this is the portion that has just been added by that synaptic vesicle joining on. Okay, and we now want to pinch that back off and return it to being a synaptic vesicle. So what then is the trigger that causes this? Well, it's believed to be the presence of the synaptotagmin proteins in this portion of plasma membrane. So remember, now that this synaptic vesicle membrane has fused with the plasma membrane, that means that the plasma membrane is now going to have the synaptotagmin proteins in. So I just draw a little synaptotagmin here. Here is the 2C2 domains that are still in the cytoplasm, and here now its amino terminus is extracellularly and its C terminus is intracellularly. Okay, so you have these synaptotagmin proteins in this portion of membrane, and this is believed to be one of the key things that triggers this portion of membrane to be re-endocytosed. Okay, right, so we'll come back to that in a moment. What then is going to bind to the synaptotagmin? That's what we want to now ask. Okay, what protein is going to come and bind here and is going to trigger the endocytosis? Well, the first thing which binds is what's known as the adapter protein complex 2, AP2 for short. So this stands for the adapter, that's the A, and then it's the protein complex 2. Okay, now this is a protein complex that consists of multiple proteins bound together, okay, and it's going to be it's an adapter protein complex because its role is to bind to the synaptotagmin, okay, and its other role then is to bind to the actual clathrin, which is going to participate in the endocytosis part of the actual pathway. Okay, so that's why it's an adapter protein, that it really is adapting the signal between this protein that is telling us to re-endocytose this portion of the membrane and the actual clathrin that's going to participate in the endocytosis. So let's have a look at the structure then of the adapter protein complex 2. And I should just say that there are other adapter protein complexes. So for instance, there's the adapter protein complex 1, the adapter protein complex 3, even the adapter protein complex 4. Okay, it's specifically the adapter protein complex 2 that's believed to be very important in Clathrin-mediated endocytosis in the synaptic vesicles. Okay, right, so let me now show you the structure of the adapter protein complex 2. So it's made up of 4 separate proteins, okay? The first one which I'm drawing at the moment is what's known as the alpha protein, the alpha subunit of the adapter protein complex 2, like so. Now, the alpha subunit is going to have 2 main portions. This portion down here, this is what's known as the body of the alpha subunit. Okay, and this portion up here, this is what's known as the ear, or more properly, the appendage, but you will often just hear it referred to as the ear. Okay, and in fact, when you look at the entire adapter protein complex 2, it does look rather like Mickey Mouse, which is why this is referred to as an ear, because it looks like the ear of the Mickey Mouse. Okay, so here in orange, this is our adapter protein complex 2. Oh, sorry, no, this is the alpha subunit of our adapter protein complex 2. So this is alpha. Okay, right. Now let's add on another subunit here. So the next subunit is going to be drawn pretty much as the mirror image of this, so I'll draw it like so. Okay, and it also has those holes there. Okay, so the mirror image of this one here, and this now is what's known as a beta 2 subunit. Okay, and again, it's split into these two separate domains where you have the ear or the appendage of the beta 2 subunit up here, and then the body of the beta 2 subunit, which is this portion down here. Okay, now those two gaps that are in the middle of the alpha beta 2 complex that we've formed so far, those are gaps for two more proteins that are going to sit there. Okay, so one of the proteins is going to be what's known as a sigma 2 subunit. Okay, so this is sigma 2, we're going a little bit more exotic with the Greek alphabet. And then finally, you've also got another protein, which I'll colour in green here, sitting in this space, and that's known as the mu2 subunit. Okay, so these four proteins, alpha, beta 2, sigma 2, and mu2, these make up the adaptor protein complex 2. Okay, and overall the adaptor protein complex 2 has this body region here, which is made up of the bodies of alpha and beta 2, along with the sigma 2 and the mu2, and then it's got the appendages of alpha and beta 2. Okay, right, so this is a complex that is involved in binding to the synaptotagmin, and then also involved in binding to the clathrin, which is going to participate in the endocytosis pathway. Okay, so which portion of the synaptotagmin then does the adaptor protein complex 2 bind to? Well, it's believed to be the C2b domain. Okay, so this second of the C2 domains, which remember binds free calciums in the exocytosis process. So here is C2b in turquoise there, and it's that portion that the adaptor protein complex 2 is believed to bind to. Okay, now another thing then that's important actually, and this is going to become really, really important later on. Adaptor protein complex 2 doesn't just bind to the synaptotagmin, it also needs to bind to PIP2 molecules in this portion of membrane here. Now remember I told you previously that synaptic vesicle membrane doesn't actually contain PI45P2, okay, which was for short PIP2. Okay, instead, the synaptic vesicle contains a lipid molecule known as PI4P. Okay, so let's just discuss what the difference between PI4P and PI45P2 is. So PI4P then, this stands for phosphatidylinositol, so you can probably predict that bit. And then it's just phosphatidylinositol 4-phosphate. So you've only got one phosphate group on this phosphatidylinositol molecule. So this is phosphatidylinositol 4-phosphate. Okay, so in the synaptic vesicles, if I draw a synaptic vesicle here, you do not have PI45P2. Instead, you have lots of PI4P. So let me just draw out the structure of PI4P, and then we'll highlight how it's different to PI45P2. Okay, so here then is the phosphatidic acid molecule, here is the inositol ring here, and now in the case of PI4P, phosphatidylinositol 4-phosphate, all you're going to have is a single phosphate group then coming off the inositol ring, and that's going to be coming off the 4th carbon there. Okay, so let's colour in the phosphate groups in vivid purple there. Okay, the inositol ring is then in blue here. Okay, and the glycerol molecule is in green. So that's the structure of phosphatidylinositol 4-phosphate here. Okay, or PI4P. This is what you have present in synaptic vesicle membranes. Now, you don't have that much PI4P present in the plasma membrane, and the reason is when the synaptic vesicles fuse with the plasma membrane, there are enzymes that are present in the plasma membrane which will diffuse into this new portion of membrane that has been added and will convert the PI4P molecules into PI45P2 molecules. Okay, so these enzymes which add a phosphate group onto the 5th the alcohol group that comes off the 5th carbon of the inositol ring here, These enzymes are what are known as PI4P, okay, 5 kinase enzymes, PI4P 5 kinase, okay, which are capable of phosphorylating PI4P molecules on the alcohol group that comes off the 5th carbon of the inositol ring, and therefore can convert the PI4P into PI45P2. Okay, so these enzymes convert PI4P, phosphatidylinositol 4-phosphate, into phosphatidylinositol 4,5-bisphosphate. Okay, like so. So this is catalyzed by this enzyme. So you have these enzymes present in the plasma membrane, and the instant you add the synaptic vesicle membrane to the plasma membrane, some of these enzymes will diffuse into that membrane and start converting the phosphatidylinositol 4-phosphate molecules into phosphatidylinositol 4,5-bisphosphate molecules. Okay, so ultimately this is a marker that this membrane, the plasma membrane, is the plasma membrane. Okay, the presence of PI45P2 in a membrane tells you that it's the plasma membrane rather than a synaptic vesicle membrane. And in fact, the adapter protein complex 2, it also binds to PI45P2 molecules, and really this is basically a checkpoint. It's basically making sure that it is actually starting this endocytosis process at the plasma membrane, because you don't want it occurring on a synaptic vesicle, do you? There is a risk that this would occur on a synaptic vesicle if it were not for this checkpoint that says, wait a second, we need to make sure this is the plasma membrane. How can we do that? Well, let's say we have to also bind to PI45P2, which is a marker of the plasma membrane. It's not in synaptic vesicles. Okay, so the adapter protein complex 2 then is going to bind not only to the synaptotagmin, but also to the PI45P2 molecules that will be present in the plasma membrane, but then not other intracellular membranes. Okay, so let's now draw this again down here. So if we have our membrane here, and we've got lots of these synaptotagmin molecules clustered together still, which are the portions which are in the portion of the membrane from the synaptic vesicle. Okay, now I'm just going to draw them very basically. We're going to have a very basic picture here. So these blobs are going to represent now our synaptotagmin molecules here. Okay, what then is going to happen is adapter protein complex 2 molecules are going to bind onto the cytoplasmic aspect of our synaptotagmin molecules. And again, I'm going to show this very basically. I'm just going to show adapter protein complex 2 again as a square, or rather a rectangle here. So this is going to represent our adapter protein complex 2, and I'll colour this in blue. And we should also remember that that also needs the PIP2 to bind, okay, which is, remember, this checkpoint that we are actually doing this at the plasma membrane. Okay, so let me colour code everything. So this is adapter protein complex 2 in blue here. Okay, in turquoise this is synaptotagmin. Okay, right. So what then is the next thing that the adapter protein complex 2 is going to bind to? Well, the next thing it's going to bind to is what's known as a Clathrin triskelion. So let's now discuss Clathrin molecules in a little bit more detail. Okay, so we'll start with how you actually create a Clathrin molecule out of a Clathrin heavy chain and a Clathrin light chain, and then we'll discuss how these things are going to assemble into Clathrin triskelions. Okay, so let's start off with the Clathrin heavy chain then. Okay, so Clathrin heavy chains are molecules that really do look like this, basically. They have these 3 important domains. Okay, so this portion over here, like so, in purple. Okay, this is known as the N-terminal domain, okay, of the Clathrin heavy chain. Okay, and I should have just made that very clear. This molecule that we're about to draw out, this is representing the Clathrin heavy chain. Okay, so up here, this is the N-terminal domain here. Okay, then this portion here, which I'll highlight up in green. So this portion here, this is what's known as the distal domain of the Clathrin heavy chain. Okay, so this is the distal domain. Then further on we're going to then have the proximal domain. And in fact, we split the Clathrin heavy chain into more than 3 domains. We split it actually into 4 domains. So then we have the proximal domain up to around there. Okay, so this is the proximal domain. And then finally, after the proximal domain, what you then have is the trimerization domain. Okay, so this here, this little portion which I haven't coloured yet, is the trimerization domain. And I think I'll colour in the trimerization domain in orange. Right, so that's the Clathrin heavy chain split into these 4 separate domains. To form a Clathrin molecule, what you have to do is join a Clathrin heavy chain onto a Clathrin light chain, and the Clathrin light chain is specifically going to associate with the proximal domain of the Clathrin heavy chain. So here in yellow, what that is representing is the Clathrin light chain, and together a Clathrin heavy chain and a Clathrin light chain make a Clathrin molecule. Okay, now, these Clathrin molecules are not actually the normal way that Clathrin molecules are assembled in the cell. Instead, what happens is they assemble into bigger structures than this. What happens is usually the form that you find Clathrin in is what's known as a Clathrin triskelion. Okay, so let me show you now the structure of a Clathrin triskelion. So basically, to create a Clathrin triskelion, what you need to do is attach 3 Clathrin molecules together. So let me show this. So here then is one of these Clathrin molecules, and I'll just colour code, well actually I'll colour code things in afterwards. Okay, now what you're going to do is you're going to attach on another Clathrin molecule like so. And basically it's the trimerization domains of the Clathrin heavy chains that are going to trimerize together. So I might just show this first. So here are the 3 trimerization domains here, like so. And then what's going to happen is this other Clathrin heavy chain will look like so, with its Clathrin light chain like so. And then this portion will come off here, again with its Clathrin light chain like so. And that's the structure of a Clathrin triskelion. And yes, it does look like a horrible symbol that reminds us of a horrible portion of history. Okay, so let's colour code the different portions in now. So here are the proximal domains in blue here. Okay, then here in yellow are the Clathrin light chains. Okay, then in green we'll have the distal domains of each of the 3 Clathrin heavy chains, like so. And then after that we have the N-terminal domains of each of the 3 Clathrin heavy chains. Okay, right, so that is the form that Clathrin usually exists in within the cytoplasm of cells, and it really does look like that. When you actually see one of these molecules for real, it does look like that. Okay, so what can now happen with these Clathrin triskelions is they are capable of associating with other Clathrin triskelions into incredible structures. Okay, they are capable of assembling into buckyball structures. Right, so let me show you, well, try to show you what a buckyball is, just in case you don't know. Okay, so basically anyone who is familiar with football, or as the Americans would call it, soccer, the soccer ball or the football, that is a buckyball, and it's that structure that Clathrin triskelions are capable of assembling into. Okay, so they're capable of making this football sort of shape, and that's really what's going to drive the endocytosis process. Okay, the polymerization of Clathrin into this sort of football-like mesh. Okay, right, so I'm going to try and show one of these. So basically, if you imagine initially the framework being in the plane of the piece of paper, so here is a pentagon, and buckyballs, the one that we're particularly going to be interested in is the structure of the chemical Buckminster fullerene, okay, where you have 60 vertices, okay, so 60 corners, and it's made by tessellating hexagons and pentagons together, regular hexagons and regular pentagons. So we'll imagine here that we have a regular pentagon, and we'll say originally that it's in the plane of the piece of paper. Now, unfortunately, this is a 3-dimensional structure, so it's difficult to show in 2 dimensions, so we're only going to be able to show a bit of it. So what you need to try and imagine is you need to try and imagine having then hexagons attached to each of the sides of this pentagon. So these are going to be regular hexagons attached to the sides of this pentagon here, and they're not going to be in the plane of the piece of paper anymore. Instead, they're going to be slanting out of the piece of paper. They're going to be coming out of the page towards us. Okay, so here are these hexagons slanting out of the piece of paper and coming towards us. So you need to try and imagine in 3 dimensions. If you try to actually draw it in the plane of the piece of paper, the hexagons wouldn't be touching, basically. If you actually did press them down against the piece of paper, then there'd be a gap in between these 2 hexagons. But if you bring them out of the plane of the piece of paper, you can make this sort of bowl-shaped structure, like so. Okay, so we've got this by putting 5 regular hexagons around the sides of this regular pentagon. Okay, and we've now got this sort of bowl shape starting to form. And then what you would do is go on in these corners that you've got here, one here, one here, one here, one here, and one here. What you're going to now do is stick the heads of 5 regular pentagons. So here is a regular pentagon going here, another regular pentagon would be going here, and these regular pentagons, they're certainly not in the plane of the piece of paper. They would nearly be vertical now. Okay, so coming out of the plane of the piece of paper towards us, which is why I'm drawing them in this weird way, because they're really coming up vertically upwards. Okay, and then you would go on around each one of these pentagons. You'd then have 5 hexagons. You've already got 2 of them in each case. You'd now extend it so that you've got 5 hexagons, and it would continue on, and basically eventually it would fold right back on itself, and it tessellates absolutely perfectly. Okay, it's one of the wonders of reality, okay, that they tessellate perfectly and form this incredible structure. Okay, now, how can Clathrin triskelions assemble into a meshwork that looks like this? Well, it's not perfect, basically. And what I mean by that is, it's not the case that you can put these together such that they don't overlap and form a meshwork like this. In reality, what happens is you get loads of them overlapping at portions of this to form this meshwork. So let me try and show you how this might work. So basically, let's show one Clathrin triskelion firstly. So maybe there's one Clathrin triskelion here. Okay, and I'll just show the heavy chains. The light chain, well, actually, no, I will show the light chains. I'll put them on. The light chains would be sitting alongside these portions here. So maybe that's one Clathrin triskelion here. Then we might put another Clathrin triskelion, which will be centered here. So let's put another one on here. So here is one heavy chain. Here is another heavy chain. And then this portion would be overlapping with that portion like so. Okay, so that's what I mean by it's not perfect. They don't form it without overlapping. You have to have them overlapping. Okay, and that's how you can assemble loads of Clathrin triskelions together. You can continue this picture on if you like, and you can then form a buckyball out of the Clathrin triskelions. Okay, so that's the important stuff about Clathrin. The basic idea then of how endocytosis is going to work, this is the crux of it all, is that by binding the adaptor protein complex 2 now to these synaptotagments, what the adaptor protein complex 2 is now going to do is it's going to bind to Clathrin proteins, and then basically you're going to start the Clathrin polymerization here, basically. Loads of Clathrin triskelions are going to start assembling, and they're going to assemble into one of these buckyball structures. And if you can imagine what that will do to the membrane, okay, if you imagine this is our membrane, and we're now starting the formation of one of these Clathrin buckyballs here, which is going to be attached to this portion of the membrane, what that's effectively going to do is it's going to bend the membrane whilst the buckyball forms. Okay, so the whole membrane is going to be bent just so that this buckyball can effectively form like so. So the buckyball formation is going to invaginate the membrane inwards, and that's the crux of this all. Clathrin is at the crux of this, basically. It's central to the bending of the membrane inwards in this way. Okay, right, so let's expand on that in a little bit more detail then now. So, it's very, very difficult to actually draw this all in one picture, so what we're going to go down to doing is representing Clathrin much, much more simply. Okay, so I'm going to now start representing Clathrin again just as a blob, basically. So if I go on to the other side, basically what's going to start happening is you're going to start by forming what are known as Clathrin-coated pits. Okay, so let's say this is the plasma membrane here, and we're going to demote the plasma membrane now to being represented as a single line rather than as a bilayer. Okay, then you'll have these synaptotagmin proteins here in this specific portion of the membrane that we now want to re-endocytose because this is our synaptic vesicle membrane. Okay, then what's happened is adapter protein complex 2s have bound to the synaptotagmin C2b domains and also to that plasma membrane lipid phosphatidylinosytyl 4,5-bisphosphate. Okay, so let's colour in these different portions. Here is synaptotagmin in turquoise. Okay, and then what colour did we previously show? Adapter protein complex 2 in blue. Okay, so, and then we've got adapter protein complex 2 in turquoise here. Sorry, in blue, rather. Okay, now what's going to happen, well, what has happened, rather, is you've started to get the formation of this Clathrin triskelion buckyball on top of all of this. So Clathrin molecules have attached onto these adapter protein complex 2s, and then you've triggered the polymerization process, okay, which is starting to form a Clathrin buckyball here. So I'm just going to represent the Clathrin molecules now, however, as rectangles just for simplicity. Okay, so the important thing is that when they polymerize, they're forming this curvature because of the fact that they are polymerizing into this buckyball mesh structure. Okay, like so. Right, and this initial thing that you are forming here, which is an invagination of the plasma membrane inwards, this is going to be called a Clathrin-coated pit. Okay, so this is called a Clathrin-coated pit because it's coated on the cytoplasmic aspect by Clathrin. Okay, so that's the Clathrin-coated pit. It is going to continue developing. It will get bigger and bigger and bigger. And what it will eventually progress to is something that looks more like this. Let's draw another picture now. So now what you'll have is an entire sort of pinched off vesicle like so. Okay, and again this is the plasma membrane. And again you'll have loads and loads of synaptotagmin molecules in here, which will all have adapter protein complex 2s bound to them. And then the adapter protein complex 2s will have the Clathrin molecules bound. Okay, so these are the synaptotagmin molecules. Then let's put on the adapter protein complex 2s, which again, I'll remind you, have bound to the C2b domains of the synaptotagmin and also to the membrane phospholipid Pi45P2. Okay, and now you've got this Clathrin buckyball really starting to take shape around here, okay, and all the Clathrin molecules will be attached to the AP2, and I'll sort of draw this all at once like so. Okay, right, now that gets a problem, however. You might think, brilliant, that's the story complete. What's going to happen is this is just going to continue on and on. You're going to finish that buckyball, and that will actually pinch off the membrane, basically. That will pinch off this vesicle, and then we finish. That's the endocytosis process finished. Not quite, basically. The Clathrin polymerization into this buckyball does not actually have the strength to finish the job. Okay, Clathrin polymerization does not release enough energy to actually cut off the membrane here and pinch the vesicle off completely. Something else is needed to finish the process off. Okay, and this final protein that actually finishes the process off and pinches this joining portion between the extracellular fluid and the lumen of this newly formed vesicle off, and this protein is going to be dynamin. Okay, so let's now study the protein dynamin. Okay, so dynamin then, it's what's known as a Bardomain protein. Okay, and the key features of Bardomain proteins is that they dimerize, and also they have a very curved structure, and their concave aspect has a positive charge on. Okay, so this is a Bardomain protein. Right, so let me show you the structure of this. So what's going to happen is dynamin proteins are going to dimerize into a structure that looks like a banana, like so. So this is made up of two separate dynamin proteins. Now near the center here, they actually have a domain that's known as the GTPase domain. Okay, and this is capable of hydrolyzing GTP down to GDP and inorganic phosphate. Okay, right, so let's colour this in. So here is the GTPase domain of each one of the dynamin dimers. Each one of the dynamin monomers. Okay, and then here is the rest of the protein. So what I'm now circling in turquoise is one dynamin monomer. Okay, now what then is the key feature of Bardomain proteins? Well, Bardomain proteins all do this. They all dimerize to form a banana-shaped structure. Okay, and on their concave aspect, which is this aspect here, this is the convex aspect, this is the concave aspect, they have a positive charge. Now what Bardomain proteins are all extremely important in is the bending of membranes, because basically this positive charge can interact with phospholipid heads, basically, which have a negative charge, and it can cause membranes to bend, basically. So, what then is dynamin going to do? Well, on this stalk that we have connecting this pinched off vesicle to the plasma membrane, dynamin molecules are going to assemble. So if this is the stalk here, what's actually going to basically happen is you're going to get dynamin proteins wrapping around this. So there's one dynamin dimer, then here's another dynamin dimer, and they're going to form spirals, basically. So you can see that it's starting to spiral, and then it will come all the way around, and then it will get back up to here, and then it will continue going around. So you get these spirals of dynamin proteins around this stalk that connects this budded off vesicle to the plasma membrane. Okay, and the dynamin proteins are connecting to this via the positive charge that is on their concave aspect, which will be interacting with the negative charge of the heads of the phospholipids. Okay, now, what then is believed to happen is once you have assembled this spiral of dynamin proteins around the stalk of this endocytic vesicle that we're starting to form here, what's going to happen is they're going to start hydrolyzing GTP down to GDP. Okay, so this is going to start hydrolyzing GTP down to GDP and inorganic phosphate, and the energy that's released by that is going to be used to basically compress this stalk, narrow this stalk. So what ends up happening is the spiral goes from being like this, okay? So if I represent the spiral just like as a line now, spiral goes from being sort of quite slack around this stalk to being like this. Okay, where it's much, much steeper now. Okay, and this compresses that stalk and narrows it down, and it's this compression and narrowing that's triggered by the dynamin helices that actually causes the breaking off of the endocytic vesicle from the plasma membrane. Okay, now, importantly, there is an example of a fly where dynamin has been knocked out, okay, and this is what's known as the Shapira mutant, okay, so Shapira mutant Drosophila. So Drosophila is a species of fruit fly that are often used for genetic experiments, okay, so if you knock out dynamin proteins in Drosophila, it produces a phenomenon known as the Shapira mutant Drosophila. Okay, and these Shapira mutant Drosophila are completely paralyzed very, very quickly. Okay, because they are incapable of endocytosing synaptic vesicle membrane back into their neurons, and therefore very quickly they run out of synaptic vesicles and they become incapable of neurotransmission, and that then leads to them becoming paralyzed. Okay, right, so that's why dynamin is so important. And in fact, if you look at the neurons from the Shapira mutant Drosophila, you find absolutely loads of these sort of structures in their axon terminals. So they're trying to endocytose membrane, but they can't complete the final step. They can't actually pinch off the synaptic vesicle from the plasma membrane because dynamin is needed to do that. Okay, so then what you will end up with then is a pinched off endocytic vesicle like so. So here is our endocytic vesicle. It will have lots of synaptotagmin proteins in. Okay, these synaptotagmin proteins will now still have the adaptor protein complex 2 proteins on top of them. Okay, and on top of the adaptor protein complex 2 proteins, you will still have the Clathrin buckyball assembled. Okay, and this now is called a Clathrin-coated endocytic vesicle that we've produced. Okay, the next important part is what's known as uncoating, which is the removal of not just the coat of Clathrin, but also the coat of adapter protein complex 2s. So you want to break off the Clathrin buckyball, and you also want to lose the adapter protein complex 2s. Okay, and that phenomenon is known as uncoating. Now, one of the key things that is believed to trigger uncoating is the conversion of the PI45P2 molecules in this endocytic vesicle membrane back to PI4P molecules. Okay, so you remember me telling you that synaptic vesicles usually do not have PI45P2 molecules within them. Okay, this is a phospholipid that is found exclusively in the plasma membrane. Synaptic vesicles usually have PI4P molecules in. However, when the synaptic vesicle fuses with the plasma membrane, that enzyme, PI4P5 kinase, converted the PI4P molecules into P2 molecules. Okay, and this was really important for the actual triggering of endocytosis, because the adapter protein complex 2s, shown here in blue, these need to bind to the PI45P2 molecules, as well as the synaptotagmin molecules. Okay, so it was very, very important in the initiation of the endocytosis cascade. Okay, but now what we need to do is convert the PI45P2 molecules back to PI4P molecules. Now, the way this is believed to occur is that when you pinch off this membrane, somehow, by mechanisms that are not completely understood at all, what ends up happening is enzymes get put into this portion of membrane. Okay, so now what's going to happen is this membrane is going to receive a dose of special enzymes, and these special enzymes are going to now be PI45P2 5 phosphatases, okay, which will cut off the phosphate group from the carbon of the 5th alcohol group of the inositol ring, and convert PI45P2 back to PI4P. Okay, and therefore you will lose the PI45P2 in this endocytic vesicle, and that will mean that the adapter protein complex 2s can no longer bind, because they needed not just the synaptotagmin but also the PI45P2, so they cleave off, and that then causes the disassembly of the Clathrin coat. So what ends up happening is you get uncoating, where this endocytic vesicle loses its coat of adapter protein complex 2s and also Clathrin, and now it will just have the synaptotagmin molecules in. Okay, and this then is our endocytic vesicle. Then what can happen is this endocytic vesicle can go off to the endosome. Okay, remember that organelle that we discussed right at the start, which is generally where endocytic vesicles that have just been endocytosed go first. It's sort of like a loading station. Okay, so they go to that endosome, the early endosome, they fuse with the early endosomal membrane, and then what can happen is little vesicles can be pinched off from the early endosome membrane, and these can then be used as synaptic vesicles again. Okay, so that now completes our discussion of synaptic vesicle endocytosis, and therefore we have completed our discussion of the synaptic vesicle cycle.